One of the leading causes of cattle sickness is BRDC, bovine respiratory disease complex. It costs cattle industry more than $1 billion annually. Today, BRDC is more difficult to combat than ever before, despite advancements in antibiotics and other vaccines. To combat BRDC, researchers began the Bovine Respiratory Disease Coordinate Agriculture Project, known as BirdCap. These researchers believe that some genes have a large enough effect on BRDC susceptibility that variants can be selected for to make a difference. It is thought that if these genes are located in the difference could be huge. Currently, there are 16.2 BRDC prevalence in feedlots, and this could be reduced significantly.
BRD is the #1 disease in our bovine species. Now to break it down, take a more.
So for this study, they screened dairy and beef animals using a GWAS, or genome-wide association study. To do this, they used SNPs, or single nucleotide polymorphisms, which are changes in nucleotide bases between different sets of DNA. So here we have a CG SNP, which forms a haplotype. Here we have a TA SNP. Here we have a GC SNP. And here we have an AT SNP. So with this, they hope to produce EPDs, or expected progeny difference measures, which will help producers select for sires that allow them to reduce BRD susceptibility in their herds.
Chromosomes contain DNA. In DNA, there are genes. So through selective breeding, we can breed out the BRDC susceptible genes. Here's an example of how we can breed out the unwanted blue and orange genes and keep all the purple good genes. Step 1, breed, baby. And then this is the DNA. So as you can see, the baby has 4 purple good pieces of DNA genes. So now our cow's all grown up and it's time to mate good old Betsy. And here's the genes. So as you can see, now we have successfully gotten rid of all the BRD susceptible genes through selective breeding.
In conclusion, BRDC is the leading natural cause of death in the U.S. beef and cattle industry. The biggest financial strain is found most commonly in high-volume shipping environments. Researchers got together to form BRD-CAP in hopes of solving this problem. The strategy behind this was the genome-wide association study as a way to identify SNPs in order to determine the location of susceptible genes.
Far across the distance and spaces between